Here, glucose can be some hydrocarbon crystals. Because most of the industries, they will utilize the hydrocarbon paste for treating these organisms. Citric acid means usually it prevents spoilage. You can hear me online. My voice is heard from there. You log in. Anyone can log in. You can hear? Because laptop is very far. Okay. Okay. See, this is glucose. Glucose is 6-carbon compound. By glycolysis, it forms 2 molecules of pyruvic acid. That is by glycolysis. Okay. So you have 2 NADH2 formation and 2 ATP formation as usual. Followed by the second step is formation of acetyl-CoA. Already familiar with this already. CoA being added, carbon dioxide being removed, and NADH2 formation. It's clear for you. Just like yesterday also we discussed these points. Acetyl-CoA can convert to acetoacetate. I will come back to it later. Let us finish this, then I'll come back to that. 2 molecules of acetyl-CoA can convert into acetoacetyl-CoA. 2 molecules. So one CoA is here. In acetoacetyl-CoA combination means 2 carbon plus 2 carbon gives 4 carbon. 4 carbon molecule is acetoacetyl-CoA. Okay. So remember, acetyl-CoA converts to acetoacetyl-CoA. Very important. 2 molecules will condense together form a 4 carbon compound called as acetoacetyl-CoA. Here, therefore, we should show one CoA being removed. Because only one CoA is there here. One coenzyme is present in the substrate. Acetoacetyl-CoA. So one CoA will be removed in the presence of an enzyme called allulase. Acetoacetyl-CoA converts to beta hydroxybutyl-CoA. Again, of course, there are 4 carbon compounds only from there. Butyric acid itself is a 4 carbon compound. Got it? So beta hydroxybutyl-CoA, how is it formed? By utilization of NADH2. Whatever NADH2 is formed in the beginning steps, they will be used here by a mechanism called reduction. Reduction occurs. Hydrogen is added to acetoacetyl-CoA to form beta hydroxybutyl-CoA in presence of acetoacetyl-CoA dehydrogenase. Beta hydroxybutyl-CoA by removal of one molecule becomes quaternary-CoA. So repeat again. 2 molecules of acetyl-CoA, acetoacetyl-CoA, beta hydroxybutyl-CoA, quaternary-CoA. First reduction, then dehydration. Dehydration occurs. Dehydration occurs to form quaternary-CoA. Quaternary-CoA converts to either butyraldehyde or choices are depending on the results. Quaternary-CoA becomes butyraldehyde. In presence of butyraldehyde, we have base. Where hydrogen is added, becomes butyraldehyde. Butyraldehyde converts to butanol. All of 4 carbon compounds. By addition of water, in presence of hydratase enzyme, it becomes butanol. So repeat again. Acetoacetyl-CoA, beta hydroxybutyl-CoA, quaternary-CoA, butyraldehyde, butanol. Or CoA removal, addition of water becomes butyric acid. They don't use them. Yesterday also. Whenever water is added, CoA removed, acid will form. Like acetic acid, butyric acid. Here CoA transfers. Then, other than this, there will be formation of propanol also. How propanol will form? Acetyl-CoA 2 molecules can also form acetoacetate by CoA removal. And by decarboxylation means removal of carbon dioxide, it forms acetone, which is 3 carbon. It's an optional if you want it. Okay, just to understand. Acetone is a 3 carbon compound. Acetone can also become one more compound called propanol, where NADH2 is used by reduction reaction. Thus, acetone dehydrogenase will forming 2-propanol. Why this is important is industrially why you can recover propanol, you can recover acetone, you can recover butyric acid, you can recover butyraldehyde or butanol by enzyme inhibitions. What they will do, they'll add specific inhibitors and block them. They will block a particular step so that you can recover only acetone. That is called as industrial applications. You'll be learning some concepts on them in industrial microbiology in the various semesters. The 6th semester you have industrial microbiology. There, how inoculum can be manipulated, how enzyme inhibitions can be useful, like that you will learn. So this is an exposure for you now in 3rd semester. What is an exposure for you? You can produce industrial products like propanol, acetone, butyric acid, ethanol, you can take up, if you are very much interested, you can take up industrial microbiology. Some universities offer food microbiology, like that, how you have industrial microbiology. They will be concentrating on all these things. Inoculum development, how to block the specific enzymatic step to recover more of only, I want only butyraldehyde, I don't want. So what I will block these enzymes or mutation by mutation. We'll see that it's only with butyraldehyde. That is your inoculum now. So your investment comes now. No need to invest again and again. So you have to recover only butyraldehyde. It make the organism produce more of only butyraldehyde and block the other enzymes. That is gene regulations and all these things. Okay, you all understood now? Butyric acid fermentation is one of the industrially important ones, and spoilage can also be considered. Whenever you have some fats, oils, they can get degraded, spoiled by butanediol fermentation, butyric acid fermentation. Fats will, butter and all will get spoiled. Butyric acid is produced. Spoilage also comes here. So this pathway is desirable as well undesirable. Okay, now you tell me how many desirable ones you have learned from yesterday. Lactic acid, homolactic acid, heterolactic acid, alcohol, propionic acid. These all are desirable. This also when you take in one way desirable, one way undesirable. Depends on the situation. Okay. But not for consumption. Understood. Industrial applications. Butanediol and methyl test mixed acid fermentations are spoilage, right? Spoilage. Like that. Undesirable fermentations. So when milk is, you'll be learning dairy microbiology in further semesters. 5th semester, you will learn dairy microbiology. In dairy microbiology, you will learn spoilage of milk. You will learn which are able to act on the fats, which will produce butyric acid, which will produce other organic acids. That time your connection of this physiology will come. But I don't find answers written by students like that. When we come to 5th semester, I'll tell you briefly. You also learned that one. When you come to 5th semester, when dairy microbiology classes are going on, or food microbiology classes are going on, nowhere the student connects the answer. Let's say some question is given in the exam. Spoilage of milk. Just telling you. They'll say, they'll write paragraphs of lactic acid formation, everything, etc. Butyric acid is formed, it will spoil the milk, protein is coagulated, everything they will write. Not one reaction, just writing lactose, arrow mark, glucose, galactose, glucose becomes acid. But physiology, touch will not be shown. You have to do that. You have to do that. Let's say you have, I'll tell you one example you will remember in your lifetime. Acetic acid fermentation is there in your syllabus here. You learn. You will be learning vinegar production in industrial microbiology. What I would say is, you practice at least, don't write everything. Write 4 substrates of acetic acid. Even if you put arrow mark, like how you write glucose, glycolysis, glucose pyruvate, at least write like that also. There's some connection of what you learned in 3rd semester with the 5th semester. So I will expect your batch to do it. Let us see. Okay, write some box and write small chart. That time automatically the examiner will conclude what? The students have understood 3rd semester connection with 5th semester. Do that. Okay, just have your book. See one simply. No need to glancing. It's all. No need to write full enzyme chapter. Nothing. Just substrate names 3, 4 right steps you write it down. That tells you that yes, this one is followed. What you should do, right? Glucose, aromatic pyruvic acid, aromatic acetal, okay? Then something that is something. One or two which you remember, you write. I know. Don't need to write everything also in the fifth sum. The third sum you remember. Fifth sum, at least some terminologies should be there. Let's say you learn alcohol fermentation. Wine will come in your industrial. What you can write there? Sucrose, aroma, glucose, aroma, pyruvic acid, acetaldehyde, alcohol. That you cannot write. You can. No? That way only you should understand how you are learning physiology related to that. You'll be learning cheese production there. In which one? In 6th semester. Sorry, in the 5th semester. The food and dairy. So when you're writing cheese production, put one box and just write glucose, then you write pyruvic acid, at least you write 1 or 2 succinyl CoA, something, then propionyl CoA. Don't need to write everything. Put arrow mark and just write something there. Then, okay, yeah, this student understood this. Online students, are you following?
Yes, sir.
Okay. Now we will shift to the next chapter, photosynthesis. So what do you mean by photosynthesis? Synthesis of carbohydrates, isn't it? By influence of light. That is called photosynthesis. We have different types of photosynthetic bacteria. Which one? Not visible. Yeah, they have this with them, no? Students, huh? Yeah. See, they have the photo with them. No need to. See, students who are online, this one is not clear for you, but anyway, you can understand. No, 1 second. I can't push it more than this. The wire is small. Not clear, huh? Yeah. They have a photo. What about the online students? Can you need this picture once again, or you have it with you? Open your phone. Open your phone. Same thing is written. Is it okay? I'll explain. Yes, that's all for time being.
Yes, sir.
If they're okay. They're okay. Lavanya, is it fine? Ashwati?
Yes, sir. Yes, sir, it's fine.
It's fine now? Okay, just understand the basics, that's all today. See, I am seeing 4 pictures. I'll go simple way. You're seeing 4 pictures here. Different pictures are there, isn't it? So one is about the purple sulfur bacteria. Second one is about the purple non-sulfur bacteria, green sulfur bacteria, green non-sulfur bacteria. Basic classification. Okay. What are these structures seen inside? These structures, what you see inside, are called as chromatophores. What are they called? Chromatophores. They will, that's a place where the photosynthetic apparatus is situated. If I ask you in plants, how is it situated? In the chloroplast. Chloroplast will have thylakoid membranes. You studied in lower classes. There is stroma. Stroma lamellae connects the grana. No? Thylakoid membranes together as folded is called as granum. So many grana are present in the stroma, liquid. This is a chloroplast, isn't it? So here there is no chloroplast because bacteria itself is a small one. Inside the bacteria will have small structures like this. These structures are called as chromatophores. Inside which it's nothing but an infolding of the phospholipid layer. It's an infolding of phospholipid layer. How are they? They have done a lot of studies. They found that they are different in the shape. For example, chromacium, purple sulfur, it has got aggregated vesicular type of chromatophores. Whereas rhodospirillum will be having spiral shaped chromatophores. Chlorobium has small club shape. and cyanobacteria or like Anabaena, they will have like thylakoid nature. They are more similar to plants. This is only group more similar to plants. How to justify more? You can justify more by seeing your table in the fourth point somewhere. They use water. Plants also use water. This also use water. Why are we stressing upon this H2S, H2S, water? You can see in your columns, H2S is written, water is written. Some of them use water, means this is the group which uses water. Even a plant will use water. They undergo a mechanism called non-cyclic Z pathway. In your PUC you have studied. Non-cyclic Z pathway is seen in plants as well as cyanobacteria. Whereas the other three don't show that at all. Okay, so what are we trying to learn here? What is photosystem? Photosystem means before the other electron carriers, I'm using the word electron carriers only again. Before other electron carriers, imagine in your mind phospholipid layer electron carriers. You imagine FMN, first one usually comes FMN, no? In other organisms. Here, photosystem. Photosystem is an important component. What is it actually? I'll just give you an idea on that. Photosystem means something which is a central pigment. Something which is central in pigment. Surrounding it, there are some other accessory pigments. Means central chlorophyll. To make it very simple for today's class. Next class we will continue again. Again I will tell you on photosystem. Don't worry about it. Central chlorophyll. There are many types of pigments, no? Photosynthetic pigments when you talk about. Chlorophyll is the main pigment. Central chlorophyll surrounded by a number of other pigments, accessory pigments, is called as photosystem or reaction center. Central chlorophyll A, main again. Remaining are other accessory pigments, xanthophylls, carotenoids, phycobilins. These are differences. Different organisms are different. Now I'll come to the point here. You see here. Bacterial chlorophyll A, bacterial chlorophyll B, carotenoids, that is written. So here you see bacterial chlorophyll C, chlorophyll A, carotenoids, phycocyanin. These all are different types of pigments. These pigments are associated with the main chlorophyll. Now, if I tell you, if it is a Anabaena, this one, which is used green sulfur, green non-sulfur. NS means non-sulfur. Green non-sulfur. Okay. I'm talking about cyanobacteria or Anabaena. They will have chlorophyll in the center. They'll have what? Chlorophyll in the center. Main pigment. Principal pigment, the word we use. Principal pigment, main pigment is chlorophyll A. Surrounding is the accessory pigments. You have heard about this word light harvesting complex, LHC and all, isn't it? The carotenoids, xanthophylls are light harvesting molecules. What happens actually in general is the light will fall on the surrounding pigments. Around 300 molecules will be there, not one or two. 300 accessory pigments will be there, completely covering. Just imagine there's a small central ball and completely many balls around. Imagine like that. That's how it looks. Okay. So photosystem looks like that, where central principal pigment will be present, accessory pigments will be present. Light falls on this. It will not go directly. Imagine 300 molecules are there like this. Imagine so many molecules are there. What will light do? Imagine like this. This is a central pigment. Light will fall onto this. It moves like a zigzag pattern. Moves like a zigzag pattern to reach that area, to reach the principal pigment. Okay. Finally, the central chlorophyll gives out energy, electron, that is called resonance energy. That is known as resonance energy. Keep these things, basics in your mind. So photosystem is present in all the bacteria. What you're seeing in the chart is naming, that's all. What is the naming? P840, P860, P880, P900. P means photosystem. PS also is photosystem. Okay. What is the meaning of 900, 890? That's the wavelength where they get activated. Nanometers. Activation nanometers. So there are a lot of differences between each and other. See here, this 860 nanometers, for example, nanospiral. Chlorobium 890. And cyanobacteria is P680, P700. This has got two photosystems, 1 and 2. That's all. Remaining one will have only one photosystem. This will have two photosystems. Which one? Cyanobacteria. That's why it's called Z pathway. Okay. They are non-cyclic. But to introduce to you one more non-cyclic is that among the pure bacteria. Why I'm using pure bacteria? These are all prokaryotic, particularly. At least this one behaves like a plant. Non-cyclic like plant. The remaining three, when you consider, now the difference I'm telling you. In remaining three, you see the board here. H2S is used by the first one and the third one. Means green sulfur, purple sulfur. Again, I'm repeating. Green sulfur, purple sulfur will use H2S. Purple non-sulfur will use organic compounds. Green non-sulfur will use water. I'll repeat once again. Purple sulfur, green sulfur will use what gas? H2S gas. That's why the name sulfur. Purple non-sulfur, green non-sulfur is there. Green non-sulfur is water, like plant. Purple non-sulfur use organic compounds, like alcohol, etc. To get the electrons. So I'm coming slowly to the topic there. So you got the differences, no? Now what difference we should learn? One more difference what we should learn. All of them will undergo light reaction and dark reaction for photosynthesis. When I'm talking about this, light reaction, keep it in mind. Light reaction means photophosphorylation. Okay. Next class, our topic is only on light reaction. Okay. Light reaction, when you talk about, there are two types in general classification, cyclic, non-cyclic. Cyclic is seen only in bacteria. Non-cyclic is seen in plants. That's what we have basic difference. But non-cyclic does exist even in certain bacteria like, see, wherever H2S is there, make it non-cyclic. They are also non-cyclic. Please, there's a mistake here. Wherever H2S is there, make it non-cyclic. In the table, you make a change here. If this, see here, in the third column, wherever H2S gas is utilized, make it non-cyclic. Tomorrow you will understand the topic of how I am telling you this. Only the organic compounds are used, no, they will show cyclic. That means only bacteria which show cyclic is Rhodospirillum. Now the question comes, what is the difference between non-cyclic of these two compared to cyanobacteria? So I'm repeating once again, Chromatium, Chlorobium, Anabena, examples. All of them will show non-cyclic. But Chlorobium and Chromatium shows the same type. Whereas Anabena shows Z pathway. I'll repeat once again. Chlorobium and Chromatium, first and third columns. We are saying H2S is used. They are non-cyclic. No, that non-cyclic is one method. Whereas this is different. This is Z. You see, underline the word Z. They are different. Tomorrow we will be learning three light reactions. Number one, cyclic for organic compounds. Number two, non-cyclic for Chromatium and Chlorobium. Number three, Z pathway for Anabena. All are clear? So three light reactions we are learning tomorrow. Followed by dark reactions. See the board down there? How many types of cycles are there? Only two types of cycles are there. C3 cycle and C4 cycle. All the bacteria like Chlorobium, Rhodospirillum, Chromatium, irrespectively, undergo only C4 cycle. Whereas only Anabena undergoes C3 cycle. Why I gave you this table is all you have to learn the basics first. Only then you will follow what you have to write for the answer. Are you clear, online students?
Yes, sir.
Okay. Pragati?
Yes, sir.
Any doubts?
No, sir, no
One
doubt.
thing, I've recorded this part today, whatever I have. Once you come to college, take it in the pen drive and listen once again if you want. Or I'll give it to Meenakshi. If you can put in the drive, fine. I'll give it to her. She'll copy it and put it in the drive. You can go through before the next class, so that you'll follow what I discussed today. Because I can't move the podium, the whole problem came. Okay. Follow then.
Sir.
Okay, sir.
Shall we do that?
Yes, sir.
Yes, sir.
Okay. Okay.
Yes, sir.
Then we'll end the class. Almost it's time up. 3 minutes are there. We'll end up the class for today. Next class we'll continue with light reaction, then dark reaction. So 2 classes we should finish photosynthesis is what I'm keeping in mind. Then we'll go with whatever. I think by the time your lab might start, exams might start. That time I'll go with online class for bioenergetics and biomolecules. Okay, let's see how things go. Okay, students, see you then. Take care.
Thank you, sir.
Attendance, yeah.
Thank you, sir.
Yeah. Somebody help her out in taking. Let her. Okay, students, shall we end the class for online also?
Okay, sir.
Okay, ma. Bye. Bye. I'm ending the class.
Sir, attendance.
Okay.